Hello, College Biology. We will be starting our third biological molecule topic, nucleic acids. So welcome to Nucleic Acids, Part 1. Instead of jumping directly into composition and polarity, we're going to actually discuss function first and then backtrack a little bit. Nucleic acids are the instructions for building proteins. Proteins are super important molecules, and so the instructions for building are also super important. We're going to talk about proteins next, so this discussion will be linked with the next discussion, proteins. Now, the polymers for nucleic acids are the functional component of this. So let's take a look. The polymers for nucleic acids are DNA and RNA. DNA is kept in the nucleus, and it is the instructions for all of the proteins that could possibly be built in an organism's body. RNA is a copy of the instructions for one protein that could be built in the organism's body. So, in the nucleus, if a particular protein needs to be made, RNA will be copied for the instructions for that protein. It will be sent out of the nucleus through nuclear pores, and it will interact with a ribosome, which will then actually read those instructions and start making the protein. So that's what nucleic acids are. They are the instructions for building proteins. So now let's take a look at the composition and structure and all of those good things. If DNA and RNA are the polymers, nucleotides are the monomers. Remember, polymers are long molecular chains made of repeating units, and monomers are the repeating units in a polymer. So this here, this series right here, that like this is linked to that, which is linked to that, which is linked to that, which is linked to that. This is our polymer in formation. So this is a zoomed in version of the things we're seeing either here in the RNA or here in the DNA. So that blue strand or this red strand is made up of a repeating thread like this. Now, each monomer in this thread, each monomer in this polymer is a nucleotide. And so that is what is being shown here, and each nucleotide is made up of multiple parts. So let's just take a look at the nucleotide as a whole for a minute. First, can we see what they are made of? Well, we definitely have carbon here. We know that each one of these points is carbon, 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 carbon. So we've got carbon. There's invisible bonds to hydrogen for each carbon. So carbon, hydrogen. We know that there's oxygen there. But this is when we start getting to add some of our other other atoms of life, which we haven't been able to use before. They also contain a phosphate group, which means we get to add phosphorus in here. And they also contain a nitrogenous base. Now, there are multiple types of nitrogenous bases. So there's actually a little bit of difference in every nucleotide depending on their nitrogenous base. And all of these are nitrogenous bases. So you can see that nitrogenous bases are essentially ringed structures, and those ringed structures contain carbon and nitrogen in the rings. So when we talk about our composition of nucleic acids, we have hydrogen, carbon, oxygen, nitrogen, and phosphorus. And since each ring can have multiple nitrogen, but each phosphate only has one phosphorus, nitrogen is more common than phosphorus. And so that's our composition. Now when we consider polarity, well, if we look at this, we can see that the oxygens over here have a negative charge. And we can see here that nitrogen is going to make polar covalent bonds with carbon, and the oxygens in the sugar are going to make polar covalent bonds. So these are very polar. There's lots of partial charges in the nitrogenous base in the sugar, and the phosphate itself has true negative charges. So nucleotides, and therefore nucleic acids, are very polar substances. Now the monomers, as we said, for nucleic acids are nucleotides. And a nucleotide is defined as a nucleoside plus a phosphate. So if we cover up this phosphate here, if we look at just the nitrogenous base in a sugar, that is called a nucleoside. When we add a phosphate to it, then it becomes a nucleotide. We're going to get into the details of nitrogenous bases and the sugars in our next video. So before we do that, let's review. Nucleic acids, their function, they are the instructions for making proteins. They are composed of hydrogen, carbon, oxygen, nitrogen, and phosphorus. They are polar. The monomers are nucleotides, and a nucleotide is a nucleoside modified with a phosphate, and a nucleoside is a nitrogenous base attached to a sugar. We're going to get into more details in the next video. I hope this helps. Struggle well.